are going to affect the plants, right? Like which we are categorizing them as biotic factors and the abiotic factors. So these factors are nothing but which are causing the stress to the plants. So you can see that a lot of the abiotic factors, and also there are certain biotic factors like the microorganisms, we can say the soil microorganisms, and there are parasites which are affecting the root system. So if you consider these biotic factors and also the abiotic factors, which is going to create lot of yield loss, or there is a crop loss which is going to destroy the feeding, or you can say that the agriculture produce will be destroyed. So in order to combat the population, right, for example, there is a statistics, right, which says that in 2050, by 2050 there is an increase in population rise like around 2.3 billions, and there is a huge challenge for this agriculture sector to combat this the nutrition values for the people who are the increase in the population. So in order to do that, we have to work really on how to look upon these stresses. Like the major cause of the crop loss is basically because of the abiotic stresses. So these are the potential abiotic stresses which we all know. So there is light, cold, heat, and also the metals and salinity. So most of the crops are basically are impacted by the drought. Okay, today's topic is on drought stress, so I would like to concentrate more on drought. So though there are other factors like the cold, heat, and metals and light, the drought plays a very important role in combating the yield losses, like in making the loss in the yield. So there are different factors which are affecting based on the drought. So like you have the stomatal closure, then there is a ROS production, then we also have the finally the cells would be dying because of am I audible, sir?
Yes, you are audible, madam.
So because of the drought which is being caused due to the abiotic stresses, so there is finally we can say that there is a shrinkage of the cell, and there is a loss in the crop. So we'll discuss more about that. So so here this table depicts that there are different kinds of stresses here. So we are since we are concentrating more on drought, so I would like to explain more on drought, the responses of the plants. So in this photograph which is present in down, we can see a lot of symptoms which are there, and as such drought is not the water scarcity exactly. It is again classified into different types, like we have severe stress, then we have mild stress, and we also have optimum drought stress. So if a plant is going to severe drought stress, then we can see these symptoms like the rolling of the leaves, and there is a scorching of the leaves. The scorching is nothing but we can call it as burning of the leaves. And there is we can also see another response with respect to flowers, like there is a sagging of the flowers, and we can also see the chlorosis, where the chloroplast will be affected, and also the chlorophyll will be disrupted. So these all impact finally the yield of the crop. So even we can see from the flowering it goes into the fruiting stage. So if all these things are not happening, because the leaves are the major play a major role as it is helping in the photosynthesis process. So if there are many studies which say that initially the root system is involved, but there are certain other studies which say that the signal of the drought is caught up by the root system. So it is a very complex mechanism. So to decipher this drought stress, so now we are going to I'm going to show you the different mechanisms. So before going into that, if you look at the data here, which is according to the statistics taken from 2000 to 2024, which was published by Feng et al. In 2024, we can see that there is a rise in the publication list. Okay, and China has published a lot of papers which are relevant to drought compared to the other countries. But looking at the steep increase in the rise in these publications, we can say that a lot of researchers are showing interest in deciphering the mechanism, and also to know how this drought can be combated. And I think this increase is because of the global warming. So finally, the global warming is leading to all these abiotic changes which are occurring because of the in the environment. And if you look at the next slide, here there is another hotspot study which was again done by Feng et al. In 2024. They have used a VOS viewer where they have identified, or this is a predicted result where they have done a hotspot analysis, and they have given certain keywords, like we can see drought tolerance, there is ABA here, and rice and other genes. And so these keywords, when they have given and then finally analyze the publication on with respect to the keywords, and these dots, we can see only the dots and the lines here, right? So these dots, it represent the prediction of the number of papers which are published. So the depending on the color of the dot, and also that is the intensity of the color of the dot, and also the size of the dot, we can say that the total number of publications is very high. Suppose if you look at this in the drought test, the size of the dot is bigger when compared to the other dots. So this we can exemplify, we say that there are a lot of publications, and these lines indicate the connection between these keywords. So this is just a hotspot data which is done by analysis through the using the computer software from 2000 to 2023. So here in this table, you can see the impact of drought stress on major crops. Like our major crops are wheat, rice, and then we have pulses. For example, if you take the soybean, which is highly rich in protein content, here we can see how the drought is impacting. It is impacting the nitrogen fixation, and there is also a decreased amount of the oleic acid content, wherein the soybean is also rich in oleic acid content. And if you look at the rice, so here the drought is impacting the leaf area, because if it is impacting the leaf area, it is going to impact the chloroplast, and thus impacting the photosynthesis process. And also the germination for seedling is also impacted because of the drought stress. Now there are different mechanisms. A plant is very intelligent, as we might not have observed. But suppose if there is any problem, humans tend to decide on what to do. But the plant doesn't do like that. So it has got its own mechanisms, where it follows three different mechanisms, like escape, avoid, and tolerate. So this is seen in with respect to different kinds of drought, as I have shown you in the previous slides, the potential different kinds of stresses. So if we talk about the drought, so here we have three different mechanisms. So out of this, the first thing, whenever the plant is sensing the drought, that is, there is any water scarcity, then it will first initially try to escape this drought, either by closing its life cycle very fast, that is, it will show up the early flowering and then it will lead to fruit set. So it will close the life cycle fast. Suppose the life cycle of a plant is, say, for 3 months, then it might, if it is experiencing a water stress, it will close the life cycle within, say, 2 months. And that is nothing but early maturity, and also it will synthesize the high amount of food material by photosynthetic process, because when it is sensing that there is going to be less water, then it tries to take up all the sugars, all the organic material which is present in the soil, and also using the photosynthetic machinery, it will try to store these organic compounds so that it will be helpful for its future. Next mechanism is tries to avoid this drought condition. It can be mild drought, or it can be optimum drought, or it can be a severe drought. So how does it avoid? Either by changing the root system. So usually the plants, they have taproot system and the fibrous root system. Then we have the primary roots, secondary roots, and the tertiary roots. So whenever the plant is sensing any drought condition, how does it try to avoid this? It will either, the primary root will go very deep inside the soil in search of water, and it will not allow the lateral roots to grow, because if the lateral roots are going further, then it will not find enough water. So and thus it will finally see that the primary root is going in depth, because it can find some amount of water. Then it will also initiate the stomatal closure. It is not like closure. It will try to minimize the stomatal pore, because we all know the stomata play a major role on the leaf surface. As the function of the stomata is, usually it helps in two ways. One is transpiration, the other function is in the gaseous exchange. So the stomata, if it is open, so there is lot of water loss. That is because of the transpiration process. So it will instead try to minimize the pore size of the stomata, so that the gaseous exchange is taking place, and also there is not much of water loss. And the next important mechanism what it follows is leaf rolling, as I showed you in the earlier photograph. That is, the plant slowly tries to roll off the leaf so that it is not exposing its leaf area to sunlight, and there is not much of the activity going on, that is photosynthesis or transpiration or respiration. And the next one is the transpiration, as I already talked about it. Now coming to the tolerance, how the plant is able to tolerate the drought stress by making various changes at the physiological level, or it can be at the morphological level, or at the biochemical level. So it tries to adjust osmotically, that is osmotic adjustment is nothing but it tries to maintain the turgor pressure, that is by exchange of the ions or either by exchange of the osmolytes, like different solutes, like we have proteins, sugars, then you have proline. So these are the different metabolites which it tries to balance out, and then that is the solute accumulation. And one important aspect which our topic is revolved on is the ABA. ABA is nothing but abscisic acid, which is a phytohormone, or it is also called as a stress hormone. So it is endogenously produced in the body, in the plant as such. So it is present at a minimal level. So whenever a plant is subjected to stress, the production of the ABA endogenously will increase, and thus there are mechanisms which fall on independence to this ABA. Of course, there are other mechanisms also, like there are ABA independent mechanisms also, which play a major role for this drought tolerance mechanisms. But ABA as such, there are a lot of reports which says that the endogenous ABA will try to make the plant tolerate the drought by by activating different mechanisms. And of course, the photosynthetic machinery will be maintained in order to tolerate the drought condition. Now, if we talk about the mechanisms for drought resistance in plants as such, endogenously, whatever is happening, if all of us might have observed that whenever there is a scarcity of water to a plant, the plant will not die immediately. Okay, so it will take some time. So first, if you just stop giving water to the plant, and we all know that there are three different stages in a plant. Like we have, like how humans have, we have young stage, adult stage, and old stage. So similarly for the plants, we have vegetative stage, then we have flowering stage, and the fruit set stage. So these are the three different stages wherein the plant requires maximum amount of water. So when during this stage, when there is water scarcity, immediately the plant will try to follow the one of the mechanisms and try to adapt itself to the water scarcity by following these mechanisms. Either it shows a change in the root system, or it can show a change in the leaf trace. Like root system, if you see here, so this is a taproot system, and these are the lateral roots. So whenever there is water scarcity, this primary roots goes inside the soil in search of water, and the lateral roots will have a lesser length. So these are all the root trace, like root volume, root surface area, and you have root biomass, which can be studied in order to, that is, this is entire thing is called as a root system architecture, where we can study based on, already it has been studied based on how the plant is able to tolerate to drought. And coming to the leaf traits, like we have different leaf traits, like leaf surface area. So whenever there is a drought condition, the leaf will try to decrease the surface area, because by doing so, whenever there is a decrease in the surface area, then as the leaf is not exposed much, so there is reduced transpiration, that is, there is reduced water loss will happen, and thus it can conserve water, and it can survive even during the drought stress. And if you look at this cell of a plant, so there are various mechanisms. So this is a healthy plant, and here you have a drought stressed plant. So if you look at the mechanisms here, there can be protein degradation, and one important mechanism is the ROS scavenging. ROS is nothing but reactive oxygen species, which are produced because whenever there is a stress to the plant. So there are certain other metabolites which are released, which can scavenge the ROS. So that is done by, again, ABA. So even the photosynthetic machinery and the mitochondria, so all these things will be affected. So whenever a plant is under the drought stress. So now our main important motto is on ABA. So how ABA is affecting the drought stress, and how it is able to make the plant tolerant to drought stress. So as we have already discussed, so there is endogenous ABA which is present in the plant, which helps in activating the different levels of mechanisms. It may be the physiological level, or it may be the biochemical level, or it can be also at the molecular level. Because of ABA, the expression of the genes will happen, and there is a stress response which is being created. So before going into the in detail about the ABA mechanism, I would just like to briefly explain the biosynthesis of ABA in plants. So here ABA, that is abscisic acids, this is we call it as a sesquiterpenoid. It is synthesized mostly in the plastics. So chloroplast is one of the plastic, as we know. So in the chloroplast, there are series of steps which take place, and it is basically synthesized from beta carotene, which is a 40 carbon compound. So this beta carotene belongs to a carotenoid family, and in the presence of Z enzyme, which is nothing but zeaxanthin epoxidase. So this enzyme will help in series of reactions to carry on in the chloroplast, and finally you get cis viloxanthin and cis neoxanthin. And there are some enzymes which are still not known here. So these enzymes will help in the conversion of this viloxanthin to cis viloxanthin, and these two intermediates in the presence of NCED enzyme. So NCED is nothing but 9-cis epoxy carotenoid dioxygenase, which plays a major role or a crucial role in the synthesis of ABA. So once this NCED acts on these two intermediates, there is a xanthoxin formation occurring in the chloroplast. So once the xanthoxin is formed, this is let out of the chloroplast into the cytosol. So the remaining mechanisms which are left out in the synthesis of abscisic acid, which occur in the cytosol. So there are two important enzymes again here, that is alcohol dehydrogenase reductase, and one more enzyme is aldehyde oxidase, which helps in the conversion of xanthoxin first into abscisic aldehyde and then finally into abscisic acid. So this is a biosynthetic pathway which occurs in the chloroplast of plants. So usually endogenously there is a lower amount of abscisic acid which is synthesized in the plants. So only during drought stress the endogenous level of abscisic acid is increased, and then it will try to enter into a signaling pathway which will help in the stress response. So here you can see the role of ABA in signaling pathway. So whenever the plant is subjected to drought and the ABA is produced, so this ABA will act as a ligand and it will bind to the receptors. There are two different pathways, as I mentioned earlier. One is ABA dependent pathway, which is occurring in the plants. The other pathway is ABA independent pathway, which also occurs simultaneously in the plant to combat or to make the plant tolerant to drought. So once this ABA is synthesized, because the plant gets a signal of drought, so the endogenous level of ABA is increased because the biosynthesis will be increased, and this ABA as a ligand will go and bind to the receptors, which are specific receptors, and these receptors will target most of the transcription factors, which are very specifically it will detect only the ABA related the receptors, and then the transcription factors will again help in combination with the cis acting elements, to increase certain gene expressions, which these genes are called as the candidate genes, because they work only specifically whenever there is a high level of ABA. So because of these gene expressions, all the downstream processing will be increased. Like there is an accumulation of osmolyte and raw scavenging can take place, and certain solutes can be formed, and lot of other mechanisms will be activated, and there can be changes at the morphological level and also at the biochemical level, which will help the plant to tolerate the drought condition. So this there are different mechanisms. Specifically, we have this ABA dependent mechanism and also independent mechanisms. In the next slide, if you can move here, so you can see here. So this is a very specific mechanism which is occurring in the plants. So whenever a plant is subjected to drought stress, so the signaling of the hormone will start, that is ABA is produced here. So then the ABA dependent pathway will keep working more efficiently. So in this pathway, whenever there is a high endogenous level of ABA recognized or signaled by the cell, the ABA will go and bind to the receptors. So these are very specific receptors, which is called as fatal RCAR receptors, which are pyrobactin resistant receptors. So this receptor will form a complex with the PP2Cs. This is nothing but protein phosphatases. So this entire complex will activate the SNRK2s. SNRK2 is nothing but sucrose non-fermenting related kinases. So usually this SNRK2s are present at a very lower level whenever there is no stress to the plant. So once this SNRK2 recognizes this complex, this will become activated, and this will in turn activate the transcription factors, that is Arab ABF transcription factors, which are ABA related transcription factors, and thus the stress responsive genes are activated, which will show up the physiological changes or the morphological changes. Because of that, the tolerance is expected in the plant. So as I said, it can be the stomata or it can be the root system. So there are actually a lot of mechanisms, but I have just briefed out only the two important mechanisms which are going on initially in the stomata and also in the root system. So we'll see how this ABA is helping in regulating the stomata under drought stress. So we know that stomata are the tiny pores which are present on the adaxial and abaxial surfaces of the leaf. So here we can see in the presence of any stress. Now here we are talking about drought stress. So also in the night time. So usually the plants tend to close the stomata. So here in many papers it is written as closed stomata, but the term is not right, I feel, because the stomata will never close. Because if it is closed, there is no gaseous exchange which is taking place, and a small amount of water should get inside so that the photosynthetic machinery is not completely activated. So here you can see whenever there is a drought stress, so immediately it is going to affect the stomata. The movements of the stomata will be disrupted. That means you can see a slight closure of the stomata. As we discussed already, that is whenever during drought stress the ABA synthesis is activated. So because of the increased level of ABA, it will go and bind to the receptors, and the PP2C ABA receptor complex is going to activate the SNRK2, and then the activation of the ABA related genes will take place, and thus the stomata will remain partially closed. Okay, and apart from this mechanism, as I said, there are many different mechanisms. The calcium also plays a very important role here in maintaining the stomatal movements, or we can say in maintaining the partial closure of the stomata. So if when this mechanism is happening, it will also help in the calcium influx. That is, the calcium will enter inside through these channels. So these are called as ion channels. So whenever there is a influx of calcium, there is an efflux of other ions, that is chloride, nitrate, and also the malate ions will go out by the use of S-type and P-type channels. So these are called as slow type anion channels and rapid type anion channels. So wherein based on these channel movements, the calcium will get inside and it will allow all the remaining other ions to go out of the guard cells. So this entire mechanism is happening in the guard cells, and there is also a potassium outflow, and also the water will go out. So because of these mechanisms which are happening in the stomata, there is a partial reduction or closure of the stomata. Thus, it will conserve the water, whatever is present in the plant, and it will not allow the plant to completely yield to the drought stress. Now, in this diagram here, you can see the opening of the stomata where there is no stress, and in the daylight, the stomata will regain its turgor. You can see the guard cells are a little bulged. That means there is water influx here, and the ABA synthesis is stopped. Here, the ABA will undergo catabolism. That means the ABA starts degrading, and there is no synthesis happening in the guard cells. And again, the calcium will go out, and all the ions will get inside by using the channels. So the water turgor is again regained back, and thus it will happily grow in the presence of water. So, but this is the mechanism which is going on in the plant to combat the drought stress. And here, these are the other mechanisms which help in maintaining the stomatal opening. So I won't go in detail about all this. So this OST1, that is open stomata gene, is very important in maintaining the turgor pressure. And there is another mechanism also. We all know water microtubules. So The tubulin protein also plays an important role because whenever the stomata is moving, like the guard cells, there is a turgor which is created, and because of the presence of the tubulin protein, the microtubules, there is a stretch and also getting back of the stomata. So these tubulin proteins, it's a recent study where the tubulins also are playing a very important role in this stomatal movement. So the other genes are also taking part here. So now coming to the root system. So initially a lot of studies were done saying that the shoot system is first initially getting affected whenever there is drought. But other study here, the taproot goes deep inside the soil so that the water scarcity is lessened, and it will try to take up the amount of water which is present around it, and the lateral root system will go down whenever there is an ADA, presence of ADA. And also this ADA will help in, as we said, that it is going to help in synthesizing certain osmolytes like sugars, then we have carbohydrates and certain proteins like proline, etc. So all these osmolytes can act as food material to the beneficial microorganisms. Thus, the rhizosphere will become very rich in these microbes, and this can help in other mechanisms which are relevant, which are related to the growth of the organism. And also this AB also helps in interacting with the AMF. AMF is nothing but arbuscular mycorrhizal fungi, which has got beneficial effect, and also it will try to lessen the drought stress. So all these factors or all these mechanisms are initiated or also activated because of the production of ABA by the plant endogenously. Now here, this slide, it shows a crosstalk mechanism. As we already discussed, the ABA is an important factor here. And the ROS production, ROS is nothing but reactive oxygen species which are produced whenever there is a damage to the cell. For example, H2O2, then oxygen free radicals. So these ROS production, so if the ROS production is happening during drought stress, then it will slowly lead to the damage of the cell membrane because it acts on the lipids. So we know that lipid peroxidation is going to damage the lipid bilayer of the cells. So whenever there is a damage to the plasma membrane, then the whole contents of the cell will come out and the cells will die, and thus the plant will die. So this raw scavenging can be done. Scavenging is nothing but removing of these raws can be done by the help of this ABA again. And also the calcium, where the CDPK is nothing but calcium dependent protein kinases, which play a very important role again. So all mechanisms will integrate together to give a plant a defense response. So all these are happening in the guard cell. So now here we can see, apart from abscisic acid, there are other hormones in the plants. As we know that auxins, gibberellins, cytokinins, there are brassinosteroids, then we have strigolactones, and we also have jasmonic acid, salicylic acid. So this APA can interact with all these hormones, and it can help in the defense response to stresses. For example, here you can see the jasmonic acid and ABA. In between these, there is a crosstalk mechanism. And as we said, that SNRK2 is the key gene which is helping in response to the drought. So by interaction with all these different hormones, still the drought stress can be combated, or any other stresses. For example, the salinity stress, and also we have the other potential stresses which can combat the tolerance mechanisms, or which can combat the respective stress. So here in this table, we can see various plant species, and also already a lot of studies have been done, and these are the genes which help in increasing the tolerance mechanisms in these plants. For example, we say petunia, then we have in rice, and a lot of studies have been done in Arabidopsis with respect to the ABA signaling and also the drought response. Now, for our understanding, I have taken few studies wherein I would like to show like how until now we have discussed how the endogenous ABA is affecting the tolerant mechanisms, and that is being affected both in the root system and also in the shoot system. So whenever there is plant takes up this drought stress, which is in different forms, it can be severe drought, it can be moderate drought, or it can be a mild drought. the plant by itself, it will never die immediately. It will try to either escape the drought, or it will try to tolerate the drought, or it will try to avoid the drought by activating different mechanisms by itself. So there are ABA dependent mechanisms and ABA independent mechanisms. So the ABA dependent mechanisms, it is being activated with a lot of transcription factors and also the response at the gene level. So there are a lot of changes which are occurring at the morphological level. For example, the root, the taproot will go in depth, and then the leaf area will be reduced, and and at the physiological level, there is an effect on the stomata, where the stomatal pore will try to partially close in order to reduce the transpiration or the water loss, and osmotic adjustments are also made inside the plant, like the production of the solutes, and also like amino acids and carbohydrates and lot of sugars like part of the carbohydrate. And it will also try to do raw scavenging, that is to maintain the permeability of the cell membrane. So now let's discuss about some of the studies wherein they have used. So once they understood how ABA is going to affect, or how ABA is going to make the plants tolerant to the drought, then they started using the exogenous ABA, that is nothing but application of the ABA from outside. Like suppose if you try to, if the endogenous level of ABA is not enough, then the plant may die or it may succumb to the drought tolerance. So in order to reduce that, the scientists have studied by giving a minimum amount of ABA, because it's not going to harm as it is just a hormone. So at a minimum amount of abscisic acid, they try to give and check if the plant is able to tolerate the drought or not. So here is one beautiful study which was done in 2010, that is by Waterland. So they have done experiments on bedding plants. So these bedding plants are nothing but the kind of ornamental plants which are marketed and which are maintained in various gardens, or they can also be maintained at the indoors to give aesthetic sense in terms of color, and also it will add up the seasonal ornamental nature for the gardens. So here they have taken 6 types of bedding plants like geraniums, petunias, tagetes, and pansies, and salvias, and marigold. So these are the plants which are not drought tolerant. So they had initially problem when they are transporting these plants, or when they are marketing these plants. So they were dying because of the lack of water, because during the transport, or whenever we shift plants from one garden to the other garden, we cannot see that totally the plant is watered or not. So they tried to study on increasing the shelf life with relevance to drought, and how to decrease the symptoms with relevance to drought. So they were able to observe some wilting symptoms and also sagging of the flowers. So they have done experiments by using 2 important methods. So the methodology of how you are giving the ABA exogenously is also very important, and it plays a very important role in the final result also. So they have used the foliar spray and also the drenching method, and by using, and they have used one concentration of around 500 mg per liter, and this is a control plant which is zero without any ABA. So which is regularly watered, and here the plant was subjected to stress, and on top of that they have given this ABA exogenously by using 2 methods, that is drench and spray. And finally they found that the 500 mg per liter of ABA, here it is written SABA, it is nothing but synthetic ABA. So it could result in increasing the shelf life with respect to all these 6 bedding plants, and it reduced the fluorosis and wilting also. So they were happily able to market these plants, and also they did not have any problems while shipping. So this is one of the interesting study. And next, coming to another study where the exogenous application of ABA was found in case of Camellia oleifera. This is an oil-yielding plant which is also called as a tea oil plant. So here again they have taken various concentrations of ABA, and the CK is nothing but the control plant which was well watered, and A0, it is nothing but the group which was sprayed with water under drought stress. And here all these 3 different categories of plants were subjected to drought stress with addition of ABA at different concentrations, that is 50 microgram per liter, 100 and 200 microgram per liter. So finally they found that ABA at 50 microgram per liter was able to give them a good result, because there was a significant change which was observed in terms of photosynthesis and oxidative stress, and they were also able to maintain the osmoticum or osmotic balance was maintained, and the plants were not dried up faster compared to the other concentrations of ABA. Now, this is another study which is taken from maize. They have done a beautiful study on maize where they have considered the control plants and also the plants under drought stress where they have not given any treatment. And then they have taken 4 different concentrations of ABA, that is 25, 15, 75, and 100. And in all these cases, they have done the amount of amino acids and malonaldehyde and carbohydrates. So here, I want to talk about this malonaldehyde, which is nothing but an indicator, or we can say a stress indicator, or we can also say that it is a drought stress indicator, which gives a very good impact on the stress, both the malonaldehyde and also the proline content. So if you subject a plant to drought stress specifically, we can see that in the leaves or the roots, the malonaldehyde content will increase very significantly. Even the proline content will also increase. Why? Because this malonaldehyde is a product of lipid peroxidation. So lipid peroxidation is nothing but whenever a plant is subjected to stress due to drought, as I said, the cell membrane is disrupted and this malonaldehyde content will increase. And also the proline, which is also called as a stress indicator, also will increase in order to make the plant tolerant, or it is a part of the tolerant mechanism. So here, when they have observed the presence of carbohydrates in control, it was normal, whereas in drought condition, the carbohydrates level was significantly lowered. Whereas when they used 75 micromoles of ABA, there was high amount of carbohydrates, which is observed actually more than the control. Why? Because the plant is trying to make a high amount of the carbohydrates in order to maintain the osmotic balance and also to avoid the drought stress. Here also you can see the amino acids. So these are acting as osmolytes. At 75 micromoles of ABA concentration, a high amount of osmolyte, significantly high amount of osmolyte was produced both in the shoot and the root, where this amino acid, specifically the proline, is going to help the plant to become more tolerant, because when when the ABA is given, it has produced more of proline, and this will help the plant to trigger various mechanisms or the various tolerant mechanisms, so that the plant can survive in the drought conditions. Even the malonaldehyde content you can see. So whenever there is a drought, this MDA content will increase because it is going to affect the cell membrane. But when ABA was given, the malonaldehyde content was remaining as normal as to that of the control, because the cell membrane was not destroyed as much as was expected in the drought condition. This is because of the ABA, which helps in scavenging of the ROS, that is the reactive oxygen species. So thus the cell membrane is intact, or it is not disrupted. So this is our study, which was part of the Ph.D. women scientist, wherein I have worked on identification and characterization of genes in signaling mechanism for drought tolerance in pigeon pea. So this is work was carried out at ICAR CRIDA with 2 of my mentors, Dr. Maheshwari ma'am and Vasudev Sarkar sir. And we have recently published a paper on this. So where we have identified, this is a combination of in silico work and also the laboratory work. So the major work is still on pipeline. So this is just part of the work which was published recently. So we have identified 12 genes which are playing a very important role with respect to ABA-mediated drought response. So after identifying these 12 genes by in silico analysis, then we have gone for the analysis at the lab. So where we selected 40 genotypes, and these genotypes of pigeon pea were varying with respect to the crop maturation. That is, we selected 14 genotypes of different crop maturation, like early duration crops, wherein the life cycle can be completed in 2 to 3 months, and then we selected 14 genotypes with a medium duration, wherein the life cycle can be increased to 4 to 5 months, and then we have selected 12 genotypes with late maturation. So altogether 40 genotypes, 40 diverse genotypes were selected to study these candidate genes. So out of the 12 genes, okay, out of these 12 genes which were selected from the in silico analysis, we took 9 candidate genes and also done the analysis on these 40 genotypes. So here we try to find out the difference with respect to the bases, like what is the difference. So here we can see individually we have tried to see for each gene, each candidate gene, which is taking part or playing an important role in signaling mechanism for ABA. So all these PP2C, SNRK2A, ABA, we have seen that these play a important role in signaling process, and this CAM is nothing but calmodulin, which is a calcium binding protein. As I showed you in the mechanism, that calcium also plays a very important role. And then we have NCD1 gene, which helps in the biosynthesis of ABA. Then the CYP gene, it is nothing but cytochrome P450 gene, which helps in degradation of the ABA. And these two genes are helping in conjugation and deconjugation pathways with respect to ABA. And we have tried to identify what is the difference. So here you can see on the gel, this is a 2% gel where we, after the PCR product was loaded, and there are very minor differences with respect to the polymorphism. So out of 12, we got only 9 polymorphic, the genes, which the 9 gene families. Out of each gene family, we have taken only one primer because of some technical difficulties. So here we can see very clearly a polymorphism, but because the agarose gel cannot detect less than 5 base pair difference, we try to use a software to find out what is the difference in terms of the base pairs. And then we identified that there is a huge difference in terms of polymorphism, like early varieties, then you have the medium genotypes and the late genotypes. So this result is in combination with all the genes, and we could see that there is a significant difference when we compare the early genotypes and the late genotypes in these genes. So with this, we can say that during the process of evolution also, these gene expression has been modified, and accordingly to the climatic conditions, the plants have been adapted respectively. So coming to the scope for future research, so there is a lot of scope for this research by manipulating chemically the signaling pathway in ABA, that is by modifying the genes which are taking part in the biosynthesis, or we can also do a modification in terms of signaling pathway, and also trying to synthesize the ABA analogs, like because as we all know that abscisic acid is a very expensive chemical, most of the experiments are cannot be done. So that is the reason it is very important to identify the ABA analogs. Already some work has been done on this, and a combination of genetic tools can help in breeding strategies or improving the breeding programs. And there is a lot of research gap with respect to this ABA related work. And ABA, apart from drought tolerance, it can also helps. It also helps in various other parameters like seed germination. Then also it helps in fruit ripening. Then it also helps in fruit set. So there are various advantages of, and also in bud dormancy. So in reducing the bud dormancy. So there are various advantages of this phytohormone. So a lot of research gap is still there. Because a lot of labs are not going for this because of the expensive nature of the chemical. And I hope that this research gap will be filled out very soon, and we will try to combat this stress, not only the drought stress, but other stresses also, to increase the agriculture production, and also to for a better survival of mankind and also the animals. So with this, I thank all the participants and also the students, and whoever has listened so patiently to my lecture. And also finally, we should all thank the plants due to which we are surviving still. So thanks a lot.
Thank you, madam. Thank you.